Hello viewers, I am Dr. Chandrakishore. I welcome you in my YouTube channel. In today's discussion, we'll be talking about Hedgehog cell signaling pathway. We'll try to understand the detailed molecular mechanism of this pathway and try to understand how this cell signaling pathway works. As you can see here, this is the protein called PTC. Don't worry for the detailed name of this. In the next slide, I have mentioned the extension of all these abbreviations that we have used in this slide. So for now, we'll not worry much about this extension of these molecules, and it will also make it easier to understand the pathway. So the left side of the panel is when there is no Hedgehog ligand, and right side of the panel is when there is the presence of Hedgehog ligand. So in the cell signaling pathway, Hedgehog is the ligand for this cell signaling mechanism. So when there is no Hedgehog, in that case, PTC protein, which is expressed on the plasma membrane, inhibits this SMO from coming to the plasma membrane. So this SMO protein, which is generally expressed on the internal vesicles, those are degraded. And why this SMO protein is degraded? Because this PTC is inhibiting this. Now, in the cytoplasm, we have something called microtubules. So in cell biology, you might have studied there are different types of cytoskeletons. It might be microfilament, it might be microtubule, or it might be intermediate filament. So there are three different types of cytoskeleton molecule, and microtubule is one of the cytoskeleton molecule. Maybe we can discuss this detail of cytoskeleton in some separate video. But in this diagram, the cytoskeleton which is of our interest is microtubules. So this is the microtubule structure, and these microtubules also has something called motor proteins. So we'll have separate discussion on this microtubule, motor, and cytoskeleton. So let us not go in much detail of this here. So we have this microtubule, and on this microtubule there is one motor protein called COS2. So this microtubule and its motor protein COS2 is associated with different types of kinases like FU, CK1, pKa, and GSK3. So these are some of the kinase molecule which are associated with motor protein which is present on the microtubule. And there are two other proteins which is Ci and SUFO. Now this Ci molecule which is associated with COS2 and SUFO, the Ci is actually phosphorylated by these kinase molecules in several steps. This kinase molecule includes Fu, CK1, pKa, and GSK3. So these kinase molecules Fu, CK1, pKa, and GSK3, these kinase molecules actually phosphorylates the Ci molecule. And when the Ci molecule is phosphorylated, the Ci molecule undergoes proteolytic cleavage, and this Ci molecule is fragmented into different fragments. The N-terminal fragment, which is actually the larger size of the Ci molecule, which we are calling here as Ci75. So Ci75 is actually the fragment, larger fragment of Ci, which is actually N-terminal fragment. And this N-terminal Ci fragment, which is Ci75, enters into the nucleus, and inside the nucleus the Ci75 actually suppress the target gene. So when there is no Hedgehog ligand, target gene is suppressed by Ci75 fragment, which is the larger fragment of Ci. Now in the right panel of this diagram, when there is presence of this Hedgehog ligand binds to the PTC receptor, and when this Hedgehog ligand binds to the PTC receptor, this PTC receptor is actually endocytosed and degraded. Now since there is no PTC molecule, so this SMO protein which is expressed on this SMO is now free to move to the plasma membrane. Now this SMO is getting expressed on plasma membrane. This SMO protein which is getting expressed on plasma membrane in the absence of PTC and in the presence of Hedgehog, this SMO protein is phosphorylated by different kinases like CK1 and pKa, and when this SMO is phosphorylated on the cytosine on the C terminal of SMO protein, then this phosphorylated SMO protein is actually stabilized. Now this stabilized SMO protein on plasma membrane, because of phosphorylation by kinases, this phosphorylated SMO protein interacts with the motor protein COS2, and this COS2 and FU both are also heavily phosphorylated by the kinase molecules, and this COS2 and FU is now engaged with SMO. Now since COS2 and FU is engaged with SMO protein, so now the CI molecule is escaped from this complex, and it also escapes the degradation, and the CI molecule can actually go into the nucleus. Because the complex which was phosphorylating and degrading the CI, this complex is now separated from the CI, and when the CI is released from that complex, this complete CI molecule without any degradation, this whole molecule of CI can enter into the nucleus. Once the CI molecule enters into the nucleus, it actually displaces the CI75, and then the CI molecule interacts with another protein called CBP, and along with the CBP, the CI molecule is going to activate the target gene. So let us have a quick recap on this. So when there is no Hedgehog ligand, PTC receptor inhibits the SMO protein from getting expressed on the plasma membrane, and this SMO protein is degraded. The cytoskeleton protein, which is microtubule here, along with this COS2 motor protein, forms a complex with different kinases such as FU, CK1, pKa, GSK3 and SUFO. This CI transcription factor is phosphorylated by these kinase molecules, and this phosphorylated CI molecule gets degraded, and larger fragment of CI molecule after the proteolytic cleavage, which is CI75, enters into the nucleus and suppresses the target gene. When Hedgehog ligand is present, Hedgehog ligand binds to the PTC receptor, and this PTC receptor is endocytosed and degraded. When there is no PTC, because this PTC is getting degraded, this SMO protein which was expressed on the internal vesicle moves to the plasma membrane. This SMO protein on the plasma membrane is phosphorylated. This phosphorylated SMO protein is now stabilized. Now this phosphorylated SMO protein interacts with the complex COS2 and FU. This COS2 and FU is also heavily phosphorylated, and because this complex is now engaged with SMO protein, so CI is now free. CI is not degraded. CI is now saved from the degradation because the complex is associated with SMO protein now. Now this complete molecule of CI without any degradation or fragmentation enters into the nucleus, displaces this CI75, it associates with CBP and activates the target protein, target gene. So in this way, the CI transcription factor is activating the Hedgehog target gene. Now you might be wondering about different abbreviations that we have used in this slide. So let us have a look on the details of those. So HH stands for Hedgehog, PTC is passed protein, SMO stands for smooth end, FU stands for fused protein, pKa is protein kinase A, GSK3 beta stands for glycogen synthase kinase 3 beta, CK1 stands for casein kinase 1, CI stands for cubitus interruptus, SUFU stands for suppressor of FU, COS2 stands for COSTL2, CI75 is actually the N-terminal fragment of CI, which is actually the larger in size. CBP stands for Crab binding activator protein. And you also might be like wondering why these names are like that, or why these names look little different. So most of these proteins were actually first time discovered in Drosophila, and in Drosophila the nomenclature system that is followed for the proteins are something similar to this. So these are some of the abbreviations for the proteins that we have used in the previous slide. So that's all for the Hedgehog cell signaling pathway. I hope you might have liked this video. If you have liked this video, please subscribe the channel, comment, and you can also give your feedback and suggestions on the channel. I thank you all for your time and patience, and we'll meet in another video with some exciting cell signaling pathway. Thank you.